Hi, Michelle Glass here, and welcome to another episode in our Chapter 10 lecture series. And we are talking about how the cell takes a DNA molecule and uses it to make proteins. Remember, there are two major processes. We're going to see transcription happening first, and we use our letter C here to help us remember that transcription occurs before translation. Transcription is the process where we convert the DNA molecule into an mRNA. So we're not going to convert the entire genome, the entire DNA molecule into mRNA. We're just going to read off one tiny section. We're going to read off a gene. A gene is our basic unit of heredity that's going to code for a protein. So let's get started. Now, transcription is obviously happening in the nucleus because that is where we find our DNA molecule. It is going to involve an enzyme called RNA polymerase, which is going to help us build that new mRNA strand. The RNA polymerase will bind to a promoter, which will help get it started. And so we can see that our first step in transcription is called initiation. And that is the point when that's the point when our RNA polymerase is going to come in and bind to our promoter sequence. Our promoter sequence here has often been described as a Tata box because you have lots of thymines and adenines there. Now notice your RNA polymerase it binds, it unwinds the helix, and it starts the process of transcribing. It's just going to be making the mRNA, copying off of one strand of the DNA. And notice that it's going to add in the 5 to 3' direction. Now, so here we can see our process of elongation. So we just see the RNA polymerase is running down the template strand of the DNA, building the RNA based on the rules of complementary base pairing. So it's pretty straightforward. Anytime there's a T in DNA, RNA polymerase is going to be adding an A. When your RNA polymerase sees an A in the DNA, is going to add a U in mRNA because there is no thymine in RNA molecule. And then G's and C's are exactly like what we saw during replication. So make sure you're looking at that and you can make sense of the rules of complementary base pairing. The template strand is the strand of the DNA that's being read by the RNA polymerase. So notice it's the strand that is going from the 3 to 5' direction so that we can just build a continuous mRNA strand, elongating from the 5 to 3' direction for the mRNA. Finally, the RNA polymerase will reach a DNA terminator sequence, and that's when it stops. That's the end of the gene. So that's when we stop building that mRNA. It separates, and then we'll see some editing takes place. Basically, what we need to be able to do is to get a template strand of DNA and be able to convert it into an mRNA sequence, and it's just simply using the rules of complementary base pairing. And remembering that mRNA has uracil instead of thymine. So pretty straightforward. The key thing here too is that that mRNA strand is not leaving the nucleus until it's edited. So that is the topic for our next video, so stay tuned.